Wonderful. Okay, so thank you again for joining. As we all know, our lives changed 20 months ago with news that an infectious and deadly virus was beginning to spread around the world. It was impossible at the time to predict all the problems that COVID-19 would cause, but I think we knew instinctively that sharing data rapidly and openly would be part of the solution. Many of us felt an obligation to take action to build data sharing solutions to help us respond to the pandemic, and that's what we're about to talk about today. Next slide. Mobilizing data sharing efforts in the middle of a pandemic is, of course, fraught with challenges, and we want to take a moment today to first celebrate the efforts of our community, to learn from each other's experiences, and to make plans to amplify the impact of shared data in the face of challenges from yet unknown variants and even future infectious diseases. I'm excited to have this discussion today with three incredible members of our community who will soon share their own stories. Our panelists include Ben Neale, co-lead of the COVID-19 Host Genetics Initiative, Melissa Haendel, who's at the helm of the National COVID Cohort Collaborative, or N3C, and Catalina Lopez-Correa of Genome Canada's CanCoGen. They'll each deliver a 10-minute presentation followed by 20 minutes of Q&A. Next slide, please. My name is Marc Fiume, and I'm CEO of a company called DNAstack. I'll set the stage with a short anecdote of our own journey working with an amazing consortium of partners to build a system for genomic variant surveillance to support Canada's response to COVID, and it's all powered by GA4GH standards. Next slide. Before the pandemic, one of the standards that we spent a lot of time on is called Beacon. Beacon's essentially a search engine for genetic variants. It allows researchers to ask the simple question, do you have samples with this genetic mutation? Yes or no? And that simple form of sharing has proven to be quite successful. There are now hundreds of beacons sharing data from millions of human genomes using this method. Next slide. So in January 2020, when COVID was beginning to spread, we immediately thought to ourselves, tracking variants is going to be important. Why don't we repurpose the standards and technologies we built for human genomes as part of GA4GH to viral genomes? Next slide. So at DNAstack, as it was with other organizations, we made the difficult decision to halt almost all of our efforts and focus on COVID. And within a few weeks, we stood up a COVID-19 beacon. And this was aggregating every openly shared genome we could possibly find, doing assembly and variant calling and enabling variant discovery over the beacon protocol. Next slide. There was immediate interest in collaboration from infectious disease researchers, from policymakers and government and cloud providers and others. And within a couple months, we formed a government-funded consortium around a common purpose to develop a system for genomic variant surveillance that was powered by GA4GH standards. Next slide. Since then, we've expanded the functionality of the platform, which is called Viral AI, to leverage a tightly integrated set of GA4GH standards for data representation, for data access, for data discovery, and analysis. And we're using the platform to index and share as many viral genomes as we can, including from NCBI and Genome Canada's VirusSeq. Next slide. The global need to share viral genomics data in light of the pandemic, I think, has in many ways accelerated the mission of our alliance. It's created awareness to the problem, an urgency to take action, the need to collaborate across boundaries and borders, and a commitment from high-level decision makers. Next slide. I'm very optimistic, I think, with progress that we've made, that we can leverage a strength as a global community that's committed to advancing sharing, to identify opportunities and challenges, and to organize and pursue a concrete steps and set of actions to help our response to COVID and set a precedent for how we can prepare for the future. With that, I'll invite our first speaker to the stage, Ben Neale. Ben is co-director of the program in medical and population genetics at the Broad, where he's also the director of genetics at the Stanley Center for Psychiatric Research. He's an associate professor in the Analytic and Translational Genetics Unit at Mass General Hospital, where he directs the Genomics of Public Health Initiative. He's also an associate professor in medicine at Harvard Medical School. And now Ben is going to tell us about a very impressive host genetics initiative that he's leading. Welcome, Ben.
Excellent. Thank you, Marc. And thank you for the opportunity to come and speak to you. Let me share my slides. Oh, 1 second. Wonderful. So thank you for that lovely introduction, Marc. It is my pleasure to talk to you about the progress that we're making in trying to map and understand the sort of human genetic variation side of susceptibility to COVID infection, to SARS-CoV-2 infection, and the severity of COVID-19. Next slide, please. So starting in March, I guess this, you might consider this an effort launched by a single tweet. So Andrea Gana, who had just taken up a position as group leader at the Finnish Institute of Molecular Medicine after completing a postdoc in my group, had kind of tried to initiate this effort to start to bring the human genetics community together around trying to understand the human genetic basis to COVID-19, both the question of susceptibility to illness and that of severity. This is all part of the sort of broader umbrella organization of the International Common Disease Alliance, which is really laser focused on exactly the same kinds of themes that GA4GH is, of how do we work together, how do we catalyze community engagement and involvement, and how do we ensure that the best possible scientific answers can be delivered for pressing human health conditions such as COVID. Next slide, please. So as part of the service to the community and part of our approach, we take a sort of big tent philosophy. So we've aggregated together a great many different cohorts. And in that process of aggregating together data from a wide variety of cohorts and collections, we've also built out resources for what different questionnaires and study designs and protocols people have used to probe and interrogate the human genetic contribution to variation in COVID susceptibility and severity. We release our results as soon as we've QC'd them and are confident in those meta-analysis results. They are freely available on the website for interpretation and use to try and follow up and understand what we're discovering. We've also expended a fair amount of effort in trying to engage the public through Twitter and blogs and social communication and outreach, with this view of, as Marc was saying, genomics is sort of jumping from a technical activity into something that's cracked the public conscience. And so there's tremendous opportunity to try and help people understand what we're learning about this illness in real time. Next slide, please. So there are sort of two main paradigms for interrogating genetic and genomic variation: sequencing and GWAS. I'll talk a little bit about the efforts for both rare variant discovery and common variant discovery through this initiative, starting with the rare variants. Next slide. So we've stood up a work group focused on aggregating whole exome and whole genome sequencing analysis and results. And we've just sort of crossed the line for the first gene discovery, replicating earlier reports in the literature in the community about loss of function mutations in TLR7 as a kind of clear risk factor for severe COVID. Next slide, please. But I think where the primary action has been, and this is, I think, a reflection of what data is in the community more generally, is really what we're learning about common variants. Next slide. So this kind of complicated and busy slide is a sort of description of where in the world all of the cohorts that have contributed analysis and results for interrogating the genetic basis of SARS-CoV-2 infection susceptibility and severity. The diamonds represent the sample size, and there are three primary phenotypes that we're studying. So the kind of left diamond in this lotus plot is focused on individuals that are critically ill with COVID-19. So the critically ill definition is that you basically needed something like intubation or ICU, being in an ICU, or potentially if you died as a consequence of of COVID, that would put you into the kind of critically ill phenotypic definition. On the right-hand side, we have just hospitalized with COVID-19. So if you were admitted to an inpatient facility because of your illness, then you were considered a case for that analysis. And then the top diamond, which represents easily the largest set of cases that we have out there in the big wide world, is just a reported positive test to COVID. And the thing about that third phenotype of the reported SARS-CoV-2 infection is that it really enabled the kind of direct-to-consumer companies like 23andMe or Ancestry to contribute alongside those sorts of, learning healthcare systems and academic health centers where I think a lot of the critically ill with COVID and hospitalized with COVID cohorts were coming through. Overall, we've got about over 2.5 million people included in the entire data set. This is, easily one of the largest, if not the outright largest, investigation of any human disease heretofore from a genetic mapping point of view. Next slide, please. So the main sorts of cohorts and approaches that we're bringing together sort of come in three primary flavors. The first are existing biobanks, so things like the UK Biobank or Decode or the FinGen project, the Estonian Biobank. Those kinds of efforts are, groups of individuals with genotype data available already, and then as the kind of phenotype information comes online, as people get COVID or are admitted as inpatient, that then gets updated and that becomes a substrate for analysis. So this is almost sort of real-time, genetic influences on the illness kinds of analyses. The second in yellow are the direct-to-consumer companies, things like Helix, Ancestry, 23andMe. They also agreed to work together and contribute, kind of setting aside perhaps some of their commercial interests in the spirit of collaboration. And then on the third side, we have these clinical studies, which are the more traditional case control kind of paradigm that we would see more typically for the investigation into any illness or disease. Most notable amongst those is probably the genomic study, is the largest single sample size contributor run by Kenny Bailey and colleagues out of the UK. Next slide, please. So for these phenotype definitions, we've got a couple of different ways of looking at, what do we call a case and what do we call a control? This lays out those criteria. I won't walk through the individual phenotypes themselves, but it's just worth noting that these three primary analyses are where we've spent most of our time and energy in trying to map and understand the basis of not only severe COVID, but also susceptibility to infection in the first place. And then the wealth of controls, it's pretty much a guarantee that there are individuals that did get COVID in the control set as well. But nevertheless, the sort of ability to leverage more information about allele frequency really powered the scans. And so we're careful in interpretation, and there is definitely some bleed and overlap between our severity analysis and the susceptibility analysis. But fundamentally, these are trying to drive it to related but not identical scientific questions. Next slide, please. So here they are, the two primary results that we think are most informative. Up top is that hospitalized with COVID-19 versus controls. We've got a bunch of loci that we've identified. I'll walk through some of those associations and how we interpret them as we go in the next few slides. But fundamentally, genetic mapping is working. Neither phenotype is particularly heritable. So that is to say, our methods for estimating the overall genomic contribution to this set of outcomes is still pretty small. But the results themselves, the top end of the results, we think are actually informative and teaching us about the potential biological pathways for both severity and susceptibility. And perhaps most strikingly, that there are loci that are specific to severity and not susceptibility, and vice versa, suggesting that there are potentially different mechanisms for proliferation of the virus within the individual versus the acquisition of an infection in the first place. Next slide, please. So here's some of the biological discoveries that we're really enthusiastic about and I'd like to highlight. The first is this IFNAR2 locus. So this is a well-known interferon signaling pathway that's involved clearly in kind of response to COVID. And that's highlighted in that kind of red box with the one down at the bottom. The red box over on the left-hand slide that's highlighting that second locus is right smack dab in the OAS gene cluster. OAS is sort of part of our innate immunity antiviral restriction enzyme activator, mechanisms. And this is, a really important component of how our body fights off viral infections more generally. And it's quite encouraging to see that we can map and identify such genetic effects so readily and rapidly through these kinds of efforts. And then the third locus that I'd call your attention to on this slide is this DPP9 association. And for all three of these genes, it is worth mentioning that we are not absolutely certain about the gene, because still the interpretation of these associations is a challenge, not just in this context, but in all contexts for human genetic inquiry. But nevertheless, the progress is there, and these associations are teaching us about places in the genome that have some material impact on risk to infection or severe illness. Next slide, please. And then following that up with another trio of associations where we have some interesting stories to tell. First amongst these is a coding variant in a gene called TYK2, which has been associated to not only a primary immunodeficiency if you have complete loss of function for TYK2, but the sort of missense variant is also that is increasing risk for severe COVID, is protective against several autoimmune diseases. And so there's very much a push me, pull you with respect to this particular association of this particular gene. And I think a lot of, kind of infectious disease human genetic investigation does suggest this sort of two sides of the coin in terms of what the consequences of given genetic associations or variants might truly be. In addition to the TYK2 association, there are associations to FOXP4, or at least the region around FOXP4. This association was only mapped through the international collaborative efforts because it is a variant that is quite different frequencies across different ancestries. And therefore, it was only through the inclusion of many diverse ancestries that we had the power to identify this in the first place. And then the final thing to touch on is this ABO association. And the association does look like it's associated to the O blood group, but it's still not super clear. It's still complicated fine mapping these things. And it is very clear, though, that it has much more to do with susceptibility to infection than it has to do with severity. Next slide, please. So that was our kind of release 5. We've just put the finishing touches on a new release, release 6. These results are all available for download as well. And there's some exciting emerging biological stories that might be coming from this updated set of results as well, including this association to SFTPD, which has been previously implicated in lung function, particularly that FEV1 over FVC, which is a measure of forced expiratory volume. And, that lung function aspect, again, gives us another clue that this might be something that's important with respect to the kind of strength of association and trying to provide a little bit more biological color. Next slide, please. There's also an association to this MUC5B, which is involved in encoding respiratory tract mucin glycoproteins. This is a well-known, strong-acting, variant for idiopathic pulmonary fibrosis, but it is protective for severe COVID-19. So again, we have another one of these push me, pull you kinds of circumstances, and I think that's going to, maybe be a kind of big story in interpreting a lot of these sorts of phenomena. And then next slide, please. And perhaps most encouragingly, we now have identified a genetic variant that's just upstream of ACE2, 69 base pairs ahead of the transcription start site, that is associated with a reduction in ACE2 expression. And ACE2 is the receptor that is thought to be the target for how SARS-CoV-2 gets into cells in the first place. And so this, I think, is a nice validation of the approach and has, I think, the opportunity to teach us a little bit about gene regulation as well. Next slide, please. So to conclude, we've identified 24 associations across severity and partial susceptibility, and it's kind of complicated. Some things are clearly loading onto susceptibility, while other things are loading a little bit more clearly onto severity, and then there's a set where it's still a little bit indeterminate. We can interpret the associations in a handful of loci, and I've really walked you through the things that we think are most interpretable and promising, but it is still the case that for most instances, we're still having a hard time understanding exactly how to interpret a genetic association. But I think most fundamental for this community is that the spirit of collaboration and working together is what made this possible in the first place. And I cannot express my humility greatly enough at the opportunity to contribute to such an important effort in the face of, I think, the defining public health crisis of our lives. Thank you.
Ben, thank you so much for that. I think you touched on a lot of themes that I would love to get back to as part of our Q&A. For now, we'll move to our next speaker, Dr. Melissa Haendel, who's, of course, a longtime contributor to the GA4GH. Melissa is the Chief Research Informatics Officer at University of Colorado Anschutz Medical School and the Director of the Center for Data to Health. Her background is in molecular genetics and developmental biology, as well as translational informatics, with a focus over the past decade on open science and semantic engineering. Dr. Haendel's vision is to weave together healthcare systems, basic science research, and patient-generated data through the development of data integration technologies and innovative data capture strategies. Melissa's research has focused on integration of genotype-phenotype data to improve rare disease diagnosis and mechanism discovery. Melissa is also co-lead of the GA4GH clinical and phenotypic data capture workstream. And now, Melissa, we're excited to learn more about N3C.
Terrific. Thank you so much. That was really a lovely introduction. It's really a pleasure to be here to talk about this particular project and all the work that our community has been doing over the past 18 months or so. So I'm going to talk today about the National COVID Cohort Collaborative. This is a community in the United States, although we do have international participants. But it really began with the Centers for Translational Science award sites, which are the 60 largest academic medical centers here in the United States, kind of putting our heads together and really thinking about what can we do to help combat the pandemic in the face of this unknown disease. Next slide, please. So obviously, we're all here to talk about data sharing in the face of the pandemic. In the United States and elsewhere around the world, it's very challenging to get your hands on large amounts of clinical data in order to do discovery analytics. And so one of the things that we really set out to do is to say, how can we actually have enough data to reveal the patterns of the clinical outcomes of this disease? What are the comorbidities? How do we personalize treatment? Which drugs exacerbate COVID outcomes? Which drugs ameliorate them? How do we distinguish between acute COVID versus MIS-C in pediatric cases? How can we predict acute COVID severity early in the course? And more recently, how do we predict, diagnose, and treat long COVID? And this has to do with really thinking about how do we use, large amounts of clinical data to test hypotheses about treatment regimes and biomarkers that can help us make better clinical decisions. And how do we actually support clinicians in all contexts, whether you're at, a first-class world-class hospital or, in a tertiary care center anywhere around the world that may be resource limited, and how do we expend those resources carefully and for the most patients that most need them. Next slide, please. So one of the most exciting things that actually happened in the context of the N3C was our data governance. And people talk about, wow, it's an amazing program that developed so quickly with so much data, and we'll get to that part too. But I actually think the most innovative part of what happened in the context of N3C was the governance. Really working together with the community, with the institutions, and with our national institutes of health and our US government, in innovative ways that have never been realized before, was really just an amazing feat and sort of dedication on part of many, many people and many institutions. So I wanted to talk a little bit about how that works. And also as a prelude to some content on the next few slides, the reason that this is so critical is because we're actually sharing the entire EHR record for these patients that we're going to be placing in this data repository. So the governance and security and ethical use of these data is really, we have to be so careful with this. And then we have our regulatory laws and our institutional regulatory oversight committees. There's just a lot of moving parts in terms of the governance and the transfer of these data for use in the pandemic context. And I know there are many other nations that are challenged in the same way. In the United States, we do not have a common electronic health record systems. So it's especially challenging to aggregate data from across different institutions. So the way it works is that institutions that contribute data have an IRB and a data transfer agreement with the government, and they thereby then transfer those data sets to the National Institutes of Health. And I should also mention that this is a fantastic partnership with some commercial organizations as well. Our repository is held in the Amazon cloud, and also in partnership with Palantir, who has created an amazing environment for doing analytics that is both secure and fully reproducible. And we'll get to that in a minute. So it's also public-private government partnerships as well. And so once the data is then delivered to NIH, the NIH has its own institutional IRB to manage the repository itself. That data is then distributed into 3 different sort of types of data in our cloud. We have a HIPAA limited data set, which contains geocodes and date stamps for service dates, which are obviously very important for understanding the spread of the pandemic and its impact. We have de-identified data that obfuscates the date shifts the dates, as well as obfuscating the zip codes to 3 digits. And then finally, we have some synthetic generated data. And this is very important because it means that we can provide the broadest possible access to the data. We have over 2,500 researchers now that have access to what is now the largest HIPAA limited clinical data set in US history. And we even have high school students and community college students able to access these data because of these sort of different provisioning levels. So as a user, and hopefully some of you will become users, you go through the process of having your institution sign a data use agreement with the NIH. You sign some community guiding principles, which essentially declare, best practices, code of conduct for interacting with the community, interacting with the system, and use of the data. Once you have a project in mind, and you must be studying COVID, you can't use these data to study anything else. You submit a data use request, which includes a user code of conduct for sort of data management and making sure that you're knowledgeable about how to use the data. And then an attestation of human subjects training and security training. So there's a lot of little hoops to jump through. But then once you do go through all of that, you go through the data access committee and get approval, and your project space is commissioned, and you can do the work on whichever tier of data that you've been given access to. For projects requiring that limited data set, you do need to have a user project specific IRB through your institution. So there's an additional layer of oversight. A lot of regulatory oversight to provide access to row level clinical data en masse. So it seems like a lot, but this whole governance structure has never been kind of realized before for this type of data. And so it's really exciting to be able to provide access to so many people to this really valuable data. Next slide. So this is a little bit about what our distribution looks like. We currently have 65 sites that have released data sets, and approximately 90 who have signed up to deliver data. There are 7.9 million patients in the system, with over 2 million positive COVID cases. And so you can kind of get a sense of the magnitude of the distribution. We've also worked very, very hard to ensure that the distribution is representative of our demographics across the United States. The dots don't necessarily fully reveal, since a dot may represent a whole region. It's just where the institution's address is on the map. But we've done a lot of work to look at the demographics, and we feel that it's representative cohort of our nation. So that is really terrific. And again, just with many thanks to all the institutions that have contributed data and who have been participating in the analytics. Next slide, please. So I wanted to talk a little bit about how we actually made this happen. So as it turns out, in many parts of the world, there are many different common data models that people put clinical data in for research. And they're, it's suggested that they're largely designed for separate reasons, but the reality is there's a lot of commonalities, and the reasons are not so clear anymore in many cases. In this case, we worked very closely with the different communities that have instantiated these different common data models: the OMOP model from the Odyssey community, the ACT model, the Trinetics as a commercial partner, again, another commercial partnership, NP-CoreNet. And then we've also been working on getting data from FHIR, although that's a longer term goal because FHIR isn't quite ready for bulk operations as yet. And what happens is the data comes in through this pipeline. We have kind of a white glove service that helps support the institution getting the data ready. It goes through a variety of parsing and data harmonization steps. We have a target. We use the target model is the OMOP model for those that are familiar. There are model mappings and value set mappings. And there's an enormous amount of quality assurance that is tracked throughout the way. This is all reproducible, so if anything goes awry, we can go back and address the issues. So in order to create this pipeline, we performed an enormous number of manual mappings and transformations, and then that was then scaled to sort of an industrial scale to manage the large quantity of data. Once all that happens, it gets all merged together into the release data set and made available in those buckets that I just mentioned. So again, this is a rather unprecedented amount of, very sensitive clinical data sharing here in the United States. Next slide, please. So I wanted to talk a little bit about, so just kind of in the spirit of GA4GH, a little bit about some of the advantages that we have learned in the creation of centralized analytics for N3C. So we talk a lot in GA4GH about distributed analytics, many wonderful projects like Beacon or Matchmaker Exchange, thinking about how do we sort of query, everyone's resources where they are. And those are wonderful things. And without those things, we too here with the N3C would not have been able to create the N3C. However, there are certain kinds of questions that you can ask in the context of having just a massive amount of patient level data that you just can't ask in a distributed context. So for example, if I want to ask questions like, how is the time on the ventilator impacted by a given drug? That question would then be sent to the distributed network partners that have their data in that data model, in any one of those data models. And then the results would be aggregated and sent back. And I could say, oh, one site said 1 day, one site said 3 days, and I could take the average or do statistics and come up with some answers. That's very different than the context of N3C, where we've centralized and harmonized all of those data, where we can ask questions using machine learning and AI techniques, such as in patients under age 60, which factors are most predictive of severe outcomes? Those are the kinds of questions that we really need in the face of a new disease. And so we've been able to really collaboratively build, test, and refine various kinds of algorithmic classifiers and identify novel associations between drugs and comorbidities in our clinical outcomes. And so, this would not have been possible without the partnership with all of the distributed networks. And so it's a really interesting kind of experiment in informatics and sort of social engineering about how do we leverage those already instantiated kind of more protected distributed networks to build this type of resource for centralized analytics. And as I'm going to talk about in a minute, it's also really critical for the kinds of data quality that we might need for machine learning and AI types of applications. And so I'm hoping that this notion inspires how we think about things in the context of GA4GH. Which projects do we actually want to try to do something similar where we might want to actually create a centralized repository from existing distributed networks? Next slide, please. So this is just a very, very simple example to get you acquainted to the challenge. So these two patients, one patient in hospital A and one patient in hospital B, both have COVID diagnoses, but a single query of data that's unharmonized would not actually capture them both. So at the top, we have a different code system used for the diagnosis. We don't even represent dates in the same way. And so it seems trivial, but it's actually non-trivial to define what does it actually mean to be COVID positive. And so there's many, many, many codes that are used to describe various kinds of COVID tests to determine COVID positive versus a diagnosis of having COVID-19. And so those kind of nuances need to be harmonized in order to be able to query to find all the patients that have a given diagnosis of COVID-19. Next slide, please. This is a much more complex example, and there are many, many different types of examples that we don't have time to go in today. But again, I just want to illustrate the sort of power of having the actual patient level data for improving the quality of the data for the types of analytical applications that we are in desperate need of in the face of this new disease. And this work was led by Richard Moffat at Stony Brook University with a large team of collaborators in helping prepare the data. And so, basically, one of the things that we've noted is that despite the fact that all of these data sources that we have from these institutions are already theoretically running all of their data quality tools over their common data model resources that they have in their research data warehouses, we found an enormous degree of variability in how things like units are represented. So, babies that weighed 100 pounds, institutions that had weight represented in both kilograms and pounds in the same field, having, how you represent pH, all of these different types of things are really not actually quality assured when they come in. And you would never know that in that distributed context if you didn't centralize the data. And so shown on the right, so actually on the left, we see the sort of original distribution of different types of units for kilograms, milligrams in the different types of laboratory assessments. And then over on the right is basically how we can actually rescue the data. So we can take a canonical unit that we can make a known conversion for where the unit is not plausible, such as the 100-pound baby. And we can actually infer that must have been a different unit. And that transformation can be applied and any outliers further evaluated or removed from the data set. And so what you see over on the right, after having done that for a variety of different labs and a variety of different units, is that the parts that are in the light green are actually the proper units to begin with, and the things that are sort of in the blue are the units that actually can be repaired. And so in this way, we basically, practically half the data that came in, even after being harmonized to a local common data model with data quality processes, was still not harmonized when brought together centrally, where we could reveal the fact that we have, this really heterogeneous landscape of how units are applied. Well, it really matters when you're doing machine learning that your units are correct. And so the ability to sort of repair data in an algorithmic way at this kind of scale has been one of the biggest lessons learned that we've had in our project. Next slide, please. So I wanted to talk a little bit about reproducibility and attribution, because again, I think this is something we've talked about for a long time in the GA4GH, and we've actually been able to realize this at a scale that is just so exciting. And part of the reason is that the N3C enclave for security purposes tracks every single action by every user, just in case we have a HIPAA breach or we have any other errors in the data that result in incorrect conclusions scientifically. And so because of that, we not only get the reproducibility, but we also get the ability to attribute every single user who contributes anything. And so what we've been working with is the contributor attribution model, which is a very, very simple model of an agent, the person or group who does something, the contribution itself, their role, what did they actually do, and the artifact that they create, such as data quality tool, a terminology, an algorithm, a piece of code. And so here's an example of Tiffany Callahan. She's a graduate student at the University of Colorado who has just graduated. Congratulations, Tiffany. And she is a curator of a very important file that's been used by many people now called the OBO to OMOP mapping file, which creates computable encodings of OMOP concepts using more rigorous semantic ontologies. And what you can see over there on the bottom is a sort of operations graph of all the different activities in a workflow. And we can attribute to each one of those who created which actions to that part of the workflow. And at the other end, we can dump out the ORCID IDs that are then associated with those contributions. And so it's a full provenance graph that shows the attributions, but also the reproducibility. This is the most transparent and reproducible system on clinical data. As you may recollect, we had many retractions early on in the pandemic because people were drawing conclusions from clinical data that no one really had access to. And so it's been really exciting to be able to build a system that has the kind of reproducibility and transparency, as well as the attribution to all of the many hands that it takes to do this work together on such sensitive clinical data at the kind of scale that we're talking about. And I think I would like to aspire that GA4GH could do something similar like this. Next slide, please. So what are some of the few exciting outcomes? So this was work that was led by Tel Bennett, and basically characterizing the cohort also led to the ability to create a machine learning model that would predict patient severity. And this was especially important early on in the pandemic when resources were scarce. How do we actually predict who's going to be the most severe, who needs ECMO, what kind of ventilator status are we going to expect, and really help hospitals plan for that. And one of the most exciting things is that the types of attributes that we found were most predictive of severe outcomes were things that could be collected in any average clinical setting, including in resource poor environments. So things like age, glucose, pH. These are really easy to collect clinical values that are collected on the first day in almost every setting. And the machine learning algorithm has high confidence, it's repeatable, it's clinically relevant, it's readily deployed, and it's easily refined. Another thing that we tested here is the fact that we were concerned that maybe it would change over the different periods of the pandemic as we had different spikes that affected different types of populations. And we found it was robust with regard to the different phases of the pandemic. So we're really excited that this type of algorithm can be used clinically around the world for helping prioritize care for patients. Next slide, please. So I wanted to talk a little bit about long COVID. And this is where I'm also hoping to inspire those that work more on the genomic side, because I think that there's an enormous amount of work to do to take what we're doing on the clinical informatics side and combine that with what's happening on both the viral and the host genome side. And this is work that Emily Pfaff has been leading at UNC. And what we've been working on is developing a machine learning model for how do we understand how do we define who has long COVID in the context of an electronic health record system. And this is really important for patient recruitment, for studies. It's really important for managing care. Does the patient have, a comorbidity or a new diagnosis that's just because they've come to the doctor more often, or is it because they actually have something that we don't yet know how to define, which is long COVID? And so and this is soon to be published. Hopefully in the next week or two, we'll have a preprint out. How do we identify? Oops, I lost my slide.
Thanks so much, Melissa. I think in the interest of time, maybe we will begin transitioning to the next speaker, but I'll let you have the
I just have one more slide. Can you just go to the next slide, please? Thanks. Actually, if I could just say really quickly, because this was the takeaway for the GA4GH, one of the things that we've been implementing is patient privacy preserving data linkage. And this is a mechanism to create data linkage between the identified patients in a clinical system like N3C with things like genomic variants like we heard about earlier. And so I think it's really important for GA4GH to think about how do we actually realize the goals of the data linkage. The genomics is amazing, but we often lack clinical data. The clinical data is amazing, but we often lack genomic data and genomic analytics. How do we bring them together? Next slide, please. And just to say thank you, the first marker paper had over 200 authors, and it took over a year to get the Clinical Informatics Journal, that's not used to having so many co-authors in this field, unlike genomics, to figure out how to index them all in Medline. So it's really, it takes a village, and it's really exciting to see this kind of team science in the context of clinical genetics and clinical care for COVID. Thanks very much.
Congratulations, Melissa. What a wonderful project, and you've clearly broken through both technical and collaborative hurdles to make an impact. I'm sure will have an impact beyond COVID itself. It's my pleasure to now introduce our last speaker, Dr. Catalina Lopez-Correa, the Chief Scientific Officer at Genome Canada. Catalina has served as Executive Director of the Canadian COVID-19 Genomics Network, or CanCoGen. She also held the position of COO at Ruta and Medellin, driving innovation ecosystems to advance social and economic development in Colombia and Latin America. Prior to that, Dr. Lopez-Correa was the CSO at Genome BC and at Genome Quebec, and held leadership positions at Decode Genetics and Eli Lilly, and has played advisory roles at the European Commission, Innovative Medicine Initiatives, and other private and public sector entities working in the application and implementation of genomics technologies. Catalina, Canada owes you a great debt of gratitude, and I'm grateful for you sharing the Canadian story today.
Thank you. Well, thank you, Marc, for this nice introduction, and thanks for the invitation to present today. So I will be talking about our initiative in Canada, the Canadian COVID Genomics Network. If I can have the next slide. So the CanCoGen network was actually created in April 2020 with an initial investment of $40 million to sequence 10,000 host genomes and up to 150,000 viral genomes. The idea with the overall project was to generate the data to inform public health and policy decisions across Canada, in each of the provinces, and across the country. Next slide. So the way we structure this project, we structure with the governance model basically was with two distinct implementation committees, one for the host side, one from the viral side, and a steering committee and a coordination committee. We thought that we had with CanCoGen a unique opportunity to try and link and make connections between the host and the viral genomes. Next slide. So it's really a project that is led by a large set of partners. We are working with a consortium of federal, provincial, regional public health authorities. We are working with academia, industry, hospitals, research institutes across the entire country. Next slide. So on the viral side, on the viral side, as I say, our objective has been to sequence 150,000. And the way we set it up, the project, was really working directly with the provincial labs. Some of them had the capacity to sequence. Some of them we have been, during this year and a half, these 20 months, we have been building that capacity in several of those laboratories. So we're working together with the genome centers across Canada and those provincial labs to increase their capacity or build their capacity so that they are able and ready to sequence SARS-CoV-2 genomes. We have developed pipelines for quality assurance, quality control, and we're working hard on making sure we share the data at a national and international level. Next slide. So one of the first success stories here with the COVID, with the SARS-CoV-2 genomic data was in the province of British Columbia, where the public health officer, Bonnie Henry, at the very beginning of the pandemic, when we started generating some of the first sequences, they started to see that there were lots of sequences in SARS-CoV-2 sequences and patients coming from the US. And that was one of the key elements and data that led to closing the border with the US. So I don't think, honestly, that, well, I have been in the field of genomics for 20 years, and I think this is the first time we see big policy decisions and big public health decisions made based on genomic data. So it's a key component of what we're trying to do, is inform policy, inform public health, and really, as we say here, take genomics out of the lab with our ability to inform those big decisions. Next slide. We have been able also in some of the provinces to trace the origins at the very beginning of the pandemic to on what the roots of the outbreaks. And we are doing really excellent job at a provincial level. So each of the provinces, like here, this paper published by Quebec, is using the data to make their decisions on a provincial level. And for those of you that are not familiar with the Canadian system, we have a federated healthcare system where each of the provinces have the autonomy to define their security, privacy rules, and the way they share the data. Next slide. So as I say, our objective was initially 150,000 sequences, and when we first decided on the 150,000, that appears to be a lot of sequences, and in reality, given the number of cases, given the amount of cases we have worldwide and in Canada, we are now actually at more than 200. We're now getting close to 220,000 sequences. So we exceed by far our target. And we have been proactively trying to share our data on GSED. We started slowly trying to get all the pipelines for QC, QA up to date, and really setting up all the process of sharing the data in Canada. And we're now at 121 sequences that are shared in GSED. And I'll talk a little bit about our VirusSeq data portal on the next slide. So one of the challenges for us too was that accessing data through GSED was problematic for some of the partners in Canada. And we didn't have access to some of the raw data we wanted to have. And we wanted also to provide an easy way to access to the data to Canadian academic partners that were ready to use the data. So in February this year, we launched this VirusSeq data portal in collaboration with actually Marc Fiume, his team at DNAstack, and Guillaume Bourque at McGill University, OACR, and partners from Simon Fraser University. And we have been slowly but surely getting the data now downloaded and working on a data flow that will allow us to simultaneously submit to GSED, submit to this portal, and potentially submit to NCBI. So next slide. Here, as I say, we have been very successful in the number of sequences. We're still having some challenges in terms of the timing to submit to GSED, to submit to the VirusSeq data portal in Canada, and to submit to NCBI. But we're clearly seeing a good trend in terms of the timing and the submissions. And it has been an absolutely incredible learning exercise for all of us in Canada, in particular for the public health agencies that are embarking into this incredible task of generating those sequences and sharing those on public repositories. Next slide. Many of you maybe saw this paper that was published in Nature Biotechnology almost a month ago, where Canada was not really, we were described as one of the outliers because we were in the cohort they made that was back, I think, in February or January this year. Our times for submissions were still very slow, 88 days in average for submission to GSED. We have been working, again, very, very hard. Our times today are around 30 days in average, and we have regions in the country that are moving as fast as 7 days in average to submit their data to GSED. So it's clearly, we're clearly moving in the right direction to improve our submission. Next slide. But we still have, again, challenges with the speed for data submission, the quality of metadata. We want to make sure we harmonize and standardize all metadata across the country, and that we really speed up the submission to international databases. Next slide. So data sharing has been at the core of what we're doing, and is an important lesson for Canada in how we're managing public health data, how are we sharing it and using it. And Cancogen has been working, as described here, at many different levels to promote and support this data sharing. Next slide. Now, on the host side, we are working in a whole different model. If I can have the next slide, please. instead of having the decentralized model with the provincial labs, we're working here with 3 sequencing centers and with CGen, the Canadian Genomics Enterprise, with 3 sequencing centers, one in Toronto, one in Montreal, one in Vancouver, that are generating those whole genome sequences and are capturing the data across the country. Next slide. I think we can go to the next. That was already covered before. Next slide. So what we're doing here is using the capacity we have built in those 3 laboratories for the sequencing, and then capturing, as we call it, this is basically a project of projects. We are capturing different cohorts that have been collected with different characteristics, long COVID, pediatric cohorts, and different cohorts that are helping us build this 10,000 COVID-19 patients cohort. Next slide. So now we have a total of 13,888 samples committed and 6,700, almost 700 samples that have been received, and a total of almost getting now close to 4,000 samples that have been fully sequenced and that have been also have all the clinical information collected. And we are now starting to get, next slide, to get requests for access. We have a data access committee that is evaluating all the requests across Canada to give access to these samples and to these data, not to the samples actually, to the data, clinical data, genomic data through our host-seq portal. Next slide. So many of you maybe saw this also publication by white paper by the World Economic Forum that is really taking on the lessons learned for the COVID pandemic and how a federated system could be key to develop this model of sharing for in times of COVID. Next slide. And that's exactly what we're trying to do in Canada. Genomics now is, as I say, out of the lab. We have been seeing an incredible traction in the way we share the data. We still have challenges and barriers, but an amazing improvement since the beginning of the pandemic on the data that we're able to share and how we're able to use that data to make concrete public health and policy decisions across the country. Next slide. And just to finish, I think COVID really has demonstrated that data literally has life or death consequences, and data sharing is at the heart of what we need to continue doing in pandemic preparedness and addressing this pandemic, the COVID pandemic now and any future pandemics. So I think with that, Marc, I think I finish here and happy to take any questions.
Well said, Catalina. And we'll now be turning to questions from the audience through Slido. But before we get there, I just want to thank each and every one of you for your contributions. I think what you've done in the face of the challenges imposed by this pandemic are truly remarkable. So thank you so much for your awesome leadership. We were previously going to go to break at 15 minutes past, but in order to give the audience opportunity to ask questions, we'll be going a little bit over time. So let's now turn to Slido. What opportunities exist for connecting host genomic data with pathogen data, and what is needed from an interoperability and standards perspective to make that happen? Catalina.
Well, I can start, and I'm sure my colleagues here will have other comments to make. But we initially, I have to say that when I joined Cancogen, I thought naively that we have under the same umbrella the host and the viral sequence, and that's going to be a super easy task to connect host and virus. It's just a matter of maybe healthcare number, and we just get the connection. And actually, nothing far from the truth in the reality that very naive thinking at the beginning. It is very, very challenging, and it's challenging not from a scientific perspective. It's challenging really from a logistics perspective, at least for Canada. And we have shared a lot of this experience with our colleagues from the US, from the UK, that are facing similar challenges in the sense that Yeah, at least for us in Canada, we collect the viral samples and we do the sequencing of the viral samples in the public health lab. And typically the samples for the human are collected at a hospital level. And there's basically no talking, no connection between those two. So what we're doing now, we have actually 5 pilot projects across Canada in different provinces that are using different strategies. Some of them are basically starting from individuals that got their viral genomes. They're contacting those and getting consent to go back and get the human genomes. So again, we are trying different approaches. We are being successful with some of them. We're learning tons, a lot on that exercise, and we're hoping to get a good percentage and proportion of the Cancogen overall cohort that is connected, but it's going to take time, and it is a real challenge.
Thanks, Catalina. Speaking of linked data, I know, Melissa, there's a world of clinical data that needs to be connected to genomics. What can the GA4GH do to help bridge that?
That's a really good question, and I think, again, I got a little heartbroken watching my other panelists and thinking about all the data that we need to do the kinds of correlations, and the way that we've been able to characterize the kinds of things like severity in many different ways that are much more granular than most of the sort of non-clinical studies have been able to. Like, how can we bring together those nuances together with the nuances in the genomic findings is really an exciting area of science, and I think really desperately needed for COVID. So really desperate to do what I call putting the patient back together again, where we take data about a patient and we ship it off to all the different places. We do all this wonderful analysis, and then we have these challenges that Catalina mentioned about reconnecting even just the viral variant data with the host genome data. But from clinical perspective, it's actually really challenging. And I think in the GA4GH, we've done a really nice job of sort of exposing very high level information about clinical data, but we haven't done a lot about like exposing much more granular content around the clinical data, especially unconsented data, like in an EHR system. And so I think, standards, both on the sort of governance side as well as on the technical side for how PPRL, the patient privacy preserving data linkage, and we have a partnership with DataVent, and no connection with DataVent, I'll just say, other than they're an amazing partner to really help us launch this. So maybe that's a discussion that we could have with them and other types of vendors that really can really facilitate those kinds of connections where you could have standards around how do you use a third party to manage the linkage between the data, and then finally just use cases that really drive that, because we, fundamentally the clinical data that we have cannot leave the enclave. It's Hotel California. So how do we do modal analytics in the context of that, sort of row level patient data not being able to leave the enclave? We can reassociate those things, but then we actually need a place to perform those analytics with the combined data. So I think there's really no standards for how you do the governance or how you deal with the setup of associating these different systems. And in the United States anyway, this has really been kind of pushing the boundaries of what's been done before with clinical data here in the US within the N3C.
Thanks, Melissa. The next question is for Ben. Ben, you mentioned that one of the sort of tent poles of the design was to create a big tent, and bringing together 150 studies is ambitious and challenging. We've experienced COVID sometimes forcibly creating opportunities and friction around collaboration between boundaries that we've never crossed before, or borders we haven't crossed before. So talk to us about how you've seen with that structure collaboration happen in ways that haven't happened before with the pandemic.
Sure. So, COVID's a worldwide problem, and, genomic solutions are not quite a worldwide solution yet. And I think that we have a lot of work to do on that on that front in particular. but throughout this whole process, there has been tremendous enthusiasm for just trying to get to the root of the problem, right? Like this isn't, people aren't being protectionist about this. No one like owns COVID research. No one is trying to jockey for prominence of position in terms of trying to make a difference in this. We share common cause because the pandemic is so bad that it really obviates many of the kind of maybe more typical, maybe I might even go so far as to say petty considerations that distract us from what our true goal should be of really understanding human health and disease much more clearly. And, a couple of months into the pandemic, maybe 4 or 5 months into the pandemic, I saw Matt might tweet, what if we treated every illness like we treat COVID? And what if we set aside our own petty interests to try and work together in this kind of way? And that's a world that I want to aspire to. And I think that's a world that the GA4GH believes in. And I think it's a world that we should try and commit ourselves to, because it's clear, it also bears worth mentioning that the notion that we would go from basically the first reported case of an illness to robust genetic associations that might teach us something about the biology of an illness within a year is even 5 years ago, that would be impossible for me to conceptualize. And so it illustrates what's possible when we work together. It illustrates what is possible when we let the science happen and we let the community engage and work with these sorts of things. And so I will completely and utterly second Melissa's call for clarifying governance structures. And then on that topic of putting host genomes and viral genomes together, I have a lot of envy for the public health epidemiologists working on viral genomics, because they take the sample, they take the barcode off, and then they sequence it. And that's okay. And we can't do that when we're working with humans. And I think that's part of the barriers that we need to overcome and build the appropriate protections and security considerations and put them in place to ensure that privacy is preserved, but still make sure that the promise of the science is actually realized.
I love that. One of the top questions that we're seeing here, I'd love to dovetail another one. It's what have we learned from this pandemic that we need to resolve to effectively face another one? And Ben, I think you showed a beautiful illustration of the importance and value of ethnically diverse data sets. And I wonder how we can leverage this moment to remember how important that is and to integrate that as part of our standard of practice going forward. Catalina, I know this is dear to your heart, so maybe you can take first crack at it.
Yes, Marc, and we see on Twitter and in so many places that actually the biggest challenge, we're concerned about data sharing, we're concerned about the sequencing, but the real challenge of this pandemic is equity. The real challenge across the board is really equity and access to the information, to the data, to communities that even vaccines, for instance, getting access to different communities to those vaccines. So with genomics, I have been monitoring what's happening in Africa, what's happening in Latin America in terms of sequencing. And well, we're fortunate in Canada. We have now 220,000 sequences. The UK has almost 1 million sequences generated. I guess one of the questions we need to ask ourselves, and I was this morning in another panel where we were asking how much is too much, maybe. Do we need to sequence all? We clearly know that we don't need to sequence all the genomes of the virus to get an understanding of how the variants of concern are moving around. And we need clearly to be able to have a global understanding of that movement of the virus. So more than 1 or 2 countries sequencing a lot, we really are at the point where we need to have a more federated or more, equitable or democratized use of this technology. I think the good news with that is that I think the sequence of the viral genomes is cheap enough and easy enough that now we're seeing many countries across the globe submitting their sequences, sequencing, learning, building the capacity, but there's still many challenges. Even thinking about consumables and reagents, when we monopolized some of those in big countries and more the rich countries, there are challenges about this equity and the access in other countries. So I think I would say Global Alliance, as we are also with Genome Canada, with G2MC and other organizations are thinking about this in a global way. I think that's the type of discussion we should be bringing, is the discussion about equity, the discussion about access, and making sure that we have a diverse population in our countries where we're sequencing. So yeah, absolutely. Many challenges, but I think also opportunities for us to learn from these and get this hopefully right at a global level and help us democratize the access to these technologies.
Wonderful. Do our panelists want to add on to that answer? Otherwise, I will ask one more question to you.
I completely agree. I mean, I think the pandemic has only pulled back the bed covers on just how deep those inequities are and how much work that we have to do. And, I think we should all look deep within ourselves about how we can advance the agenda of building a more equitable society.
Wonderful. Okay, picking off our last question here. We'll ask this question to Ben and also Melissa. It's great to see that DTC, direct to consumers, were willing to set aside commercial opportunities to collaborate. You both have collaborated with commercial parties and even Genome Canada, and this may be a new experience. So any thoughts and advice on how research groups can continue to engage with these types of companies in a more sustainable way?
It's a great question. I mean, each of these direct to consumer companies have their own approach to how they think about, primary genetic discovery, whether that's in their remit or not, and the rules that govern how to engage. There are research portal there's a research portal for applications with 23andMe that you can file and request, but they're going to pick and choose in the way that they're going to pick and choose. I think another company that also certainly did the right thing was Regeneron offering to generate data on samples that were collected but didn't have data generated. Thermo Fisher, formerly Affymetrix, did the same thing. I think Illumina has done a fair amount in terms of trying to support and underwrite the kind of sequencing efforts. Like the corporate sphere has kind of heard the call that society put out to address the pandemic. And I guess I just kind of want to attune them to doing that more generally, right? It doesn't have to be this severe for them to be motivated to move in this direction. There are a few other things that we could contemplate in terms of like embargo periods or kind of clear guidelines to how to structure such partnerships, because it can be challenging. And it's obviously not something that everyone's naturally going to do in terms of their research enterprise. And so I think ways of figuring out what those partnerships should look like, what the kind of typical approaches to sharing can and cannot be, and how long embargo period should be, and all of those kinds of details are the sorts of things that I think slow down this kind of collaborative enterprise. And so maybe a little bit of standardization and consistency about what good guidelines and starting points for that might be, and how to promulgate those across the community more broadly might facilitate a deeper integration between the academic and industrial sides of the equation, trying to learn about human health and disease.
Yeah, maybe I could add a little bit onto that, because I think, and I completely agree with Ben, although our engagements have been a little bit different in the sense that, what we've needed is production level infrastructure, in a hurry, but also customizable. Like it's not something that anybody had ready out of the box, but without some of those, kind of just, development teams at the ready in those, commercial organizations to build production quality software, really quickly from existing, efforts, I think was really key to the instantiation. But also just the sort of scientific partnership in creating that in a fit for purpose manner. One of the challenges that we've had, and I would urge anyone who's interested in the audience to please contact me, is we've actually been really challenged at engaging pharma and other kinds of commercial organizations because the data is hard to use. They don't really want to come in and just use the data. They want to be delivered answers. They have analysis questions, and they need people in our community to go and do them for them. We don't really know how to do that. And I think that's another kind of example that maybe GA4GH could take a lesson from, is that nobody in the commercial pharma community is going to use what we're building. But what we can do is create, a community generated service center of some kind to create those partnerships so that the experts in our systems can actually deliver the right answers for the right reasons in the right contexts. And so I think that's still an ongoing challenge for us in terms of, validating a lot of our drug discoveries and things like this. So that might be something that GA4GH could also help with.
Wonderful. That's a great segue to the next session, which is on building bridges to industry and clinical care. So I want to thank our speakers again, Ben, Melissa, Catalina. So admire your work, and thank you to those who have joined us. So I'll now pass to Lindsay Smith to lead us to the next session.